Hi again! So previously, we learned about what gene therapy is as well as its benefits. So now we'll learn about the conditions required for gene therapy. So how does gene therapy work in general? Let's just give you a whole overview of how it works. So first, we need to identify the therapeutic gene, i.e. the gene that we want to add to correct the genetic defect. Isolate this gene and link it to a promoter to ensure the expression of the transgene. The transgene is basically a gene or genetic material that is to be transferred from one organism to another. Then either of two steps can occur. The first method shown involves a direct transfer of DNA into the cells that need correcting. The introduced genes then express the gene product that was previously defective or lacking. The second method, also known as the indirect method, involves the transgene being inserted into a vector, such as a virus or a liposome, and then this leads to the genetic material being introduced into the cells that need correcting. Following that, the introduced genes just express the desired gene product. So, before gene therapy can be used in humans, some conditions must be met. There are 5 requirements in total. Firstly, the gene to be transferred must be physically available. This means that the gene must have been sequenced and isolated. Secondly, an effective method of introducing the gene into human cells is necessary. These include, and are not limited to, liposomes, gene guns, and viruses. Thirdly, the target tissue or target cells must be accessible to the gene transfer technique. Fourthly, gene therapy must not harm the patient. As the technique is still quite new, gene therapy does carry a high risk in some cases. And lastly, gene therapy currently is used as a treatment of last resort. There must be no other form of effective therapy available before gene therapy can be used. Now, let's move on to how gene therapy is implemented. There are quite a few strategies available, but we will just learn about one. The strategy that we will cover is known as gene augmentation therapy. This involves the insertion of a normal functional allele into a non-specific location within the genome to overcome the effect of the non-functional alleles. As mentioned previously, gene therapy is usually used to treat recessive disorders. It isn't usually used to treat dominant disorders, as dominant alleles causing disease usually do not code for inert or inactive products. They usually code for products that actively disrupt metabolic processes and directly affect the patient's welfare. And so, to do that in the case of dominant disorders, the defective allele must first be shut down, and then gene augmentation then is performed. And this is really hard because mastery of gene augmentation is not too great yet. So, as we mentioned briefly in the previous lecture checkpoint, gene transfer can occur either ex vivo outside the body or in vivo inside the body. For the ex vivo approach, cells are first removed from the body. Cells chosen are usually those that are easily removed and reinserted. These cells are then cultured in vitro for a period of time to increase the cell number. Normal copies of the gene are then transferred into cells in vitro, done either directly or via vectors such as liposomes or viruses. These genetically correct cells are then returned to the patient. Gene therapy in this case is worthwhile only if these cells can persist and function after transplantation. For this ex vivo approach, cells must be responsive to culture and must be able to grow outside of the body. This approach uses the patient's cells, hence ensuring no adverse immune responses. It works well with blood disorders as removal and reintroduction of bone marrow stem cells is easy. And because stem cells are used, it's a long-term solution. For the in vivo approach, this involves the transfer of genes directly into the cells of the body. There must be sufficient number of cells available for transfection, as well as a suitable gene delivery system to target the correct cell type. The in vivo method is advantageous as success is not dependent on the ability to culture cells in vitro and the subsequent transplantation of these cells into the patient. So, at this checkpoint, you should be able to talk about the conditions required for gene therapy to occur in humans, as well as the two approaches in which gene transfer for gene therapy purposes can take place.